Bene, è giunta quell'ora. Buonasera a tutti.
Very well. Welcome. Good evening to all of you. And I'd like to thank in particular the three speakers who are so qualified and who have joined us to discuss this invisible world. This is an important conference for us because 3 years ago, we started this fight, this battle. Maybe saying battle is a bit too much, but it's a kind of battle for cheeses, natural cheeses without industrial starters. And our goal is to be able, maybe at next cheese or the cheese after next one, to only showcase and display only natural cheeses. So at cheese now we have only cheeses made without pasteurized cheese, except for 2 exceptions. But we would like to be able to showcase only natural cheeses with no, let's say, external or foreign additive. We started this battle 3-4 years ago. We are at a tough moment right now because, it's pretty simple to discuss among experts, and they agree. And however, it's much more complicated to talk about this with consumers because as far as raw milk is concerned, well, people are now have now understood the difference, and we also organized a tasting, and we tasted the same cheese made with raw milk and made with pasteurized milk. And 90% of consumers have identified and said that the one made with raw cheese is better. However, with starters, it's a little bit more complicated because it's hard for people to understand this invisible world that is making this. And it's even difficult to classify these microorganisms as animals. And so if you think of bacteria and fungi and spores, and you think that they are animals like lions and whales, that's strange, you know. So for consumers, it's also almost impossible to get this vision. So as to this invisible world, we, so ordinary citizens, so we have an attitude that is a bit based on mistrust and fear. So we're fearful, and this is also due to microbiologists who talk about these things only when there is some disaster, some problem, some contamination, some disease. And COVID aside, of course, but if there is a listeria on a cheese, and then this invisible world is discussed and said to contaminate everything. But the good part about this invisible world, which is the prevalent one, the prevailing one that enables us to enjoy beautiful fermented food, it's a huge universe of fermented products and bread and cheese and wines, etc. Then this is less noticed, it goes unnoticed. And so people tend to have an attitude of mistrust. They don't trust this thing. And we would like to overcome this distrust. And we would like to tell people listening to us and people who come at cheese that this invisible world makes cheese become what it is. Of course, well-made cheese, not cheese with industrial starters. That's a completely different story. That's something else. And therefore, to discuss about this topic, so these invisible world, because it's very hard to grasp that. It's very difficult for people to understand. But we have invited 3 scientists, or actually 2 scientists and 1 producer who every day fights with this invisible world, to tell us what is your opinion about this. And now I'd like to leave the floor to Mother Noella Marcellino, whom I thank so much for participating in this conference. And she is a prioress of the Our Lady of the Rock Benedictine Monastery and a microbiologist. And she began making cheese cheese back in 1977, and in particular the Regina Lautis Bethlehem cheese. So in a place that is extremely important for microbiology, because this trust there is even more, I mean, in the States, more than here. So I leave the floor to Mother Noella. You have the floor.
Thank you. I am sharing my screen so you can see my slides. Thank you very much. Now, this is the cow who got me into microbiology and cheese making. Her name is Sheba. And when we first got her in the 70s, we discovered we had to make cheese because that is a great way to store milk. And I did not know how to make cheese, so I was praying that an old French woman would come and teach me how to make cheese. Instead, it was a young French woman who came. She was from the Auvergne, and her grandmother taught her how to make Saint-Nectaire. So we spent 2 days together making cheese using a very traditional technique, using a wooden barrel, and eventually she sent me forms from the Auvergne and a cheese paddle. Of course, we could not make Saint-Nectaire in the United States. That is a protected cheese in France, and we could never come close to that wonderful cheese. So we call our cheese Bethlehem cheese. So it's very important to note that we never use commercial cultures. When we're making the cheese, we do not use lactic acid cultures. We also do not use commercial or industrial cultures of fungi. So this cheese is a surface fungal ripened cheese, and it is produced naturally. And I noticed in the days of watching the cheese age in our little cellar of a house that the succession of microorganisms growing on the cheese were reproducible and predictable, again, without ever adding commercial strains. So when I was sent off to the university to study agriculture, I ended up in microbiology because I was making a surface ripened cheese. And so what we wanted to do was to see if we could visualize the ripening process in situ on the cheese rind and in the cheese curd, and to see if we could see these populations developing. So we used scanning electron microscopy and light microscopy. And we started with the cheese. This is cheese at day 0. So there's nothing growing on it. It would be rubbery and tasteless. So under the electron microscope, this is magnified 500 times. You can see a sort of blank surface on the top of the cheese. Nothing is growing there. And what we see this with the light microscope, there's no growth on the top. And then what we see are 2 isolated pockets of lactic acid bacteria. The lactic acid bacteria are responsible for, I'm sorry, but my at the moment, we are not able to see my pictures. They're not showing up. On day 2, what we see is the cheese is covered with a lawn of microorganisms. And what that is are yeasts. And for some reason, they are not showing up at the moment. But on day 7, what we have is a furry coat on the cheese, and that is due to the zygomycete mucor. Now, these are the yeasts. We had to go back. What the yeasts do is they metabolize lactic acid produced by the lactic acid bacteria. They start raising the pH, and they contribute vitamins. On day 7, we have the mucor, which is a zygomycete fungus. And what we see on top there are the sporangia. Those are the reproductive structures. Every hair on that cheese has this round ball with thousands of spores on it. And here by day 9, the cheese is covered with a gray furry coat, and that is due to the spores breaking off of the sporangia. And on the right, we have what we call hyphae, vegetative hyphae, and those are due to Geotrichum candidum, which is a yeast-like fungus that is growing down into the surface of the rind, sort of aerating the surface. And this is what became the subject of my doctoral dissertation, was Geotricum candidum. It's found on Brie, Camembert, Tome de Savoie, Saint-Nectaire. So it's a ubiquitous yeast-like fungus, very important in the breakdown of protein and lipids. So my cheese master came back after 2 years and noticed we had this pink fungus. She called it the flower of the molds. And that is due to Trichothecium roseum. That's two-celled conidiospores that we see on the right. And this is a mold that the cheesemakers wanted very much on their cheese. So by day 37, what we have is a dry surface, but underneath we have Brevibacterium lineans. That is the bacterium that makes the cheese surface orange, as in Limburger, Taleggio, Munster. And that creates that wonderful smell in cheese. Now, Brevibacterium needs a higher pH to grow, and also it is fairly salt tolerant. So the previous populations have prepared the cheese environment for the population that comes afterwards. All this is happening within 2 millimeters of a cheese rind. We have the lactic acid bacteria, we have the yeast, then we have mucor, Geotricum, Brevibacterium. So I wanted to see if I could study further the native strains, the natural strains of microorganisms in cheese caves in a traditional setting. I was lucky enough to get a scholarship to go to France and especially study in 6 regions, the traditional caves. You might say, why would one put a cheese, a food product in the earth, in a cave? Well, that's because 1 gram of soil can harbor up to 1 billion bacterial cells and up to 1 million individual fungal species. So these fungi and bacteria are present in the environment, in the earth, and they are what contribute to the consistency and flavor development of a cheese. We benefit from their metabolism. And the 3 processes, the biochemical processes in cheese ripening, fermentation, we talked about lactose being converted to lactic acid. Then there's the breakdown of lipids to fatty acids and glycerol, and proteolysis, which is the breakdown of protein, probably the most important biochemical process in cheese making. So this is one of the beautiful caves in France, the caves of Saint-Julien in the Auvergne. So we'll take a little visit there. When I first went to this cave, Alphonse Ballant, whose family has made Saint-Nectaire for many generations, when we first went into this cave and you see many Saint-Nectaire on rye straw on the floor of the cave, he said to me, the regulators and inspectors cannot kill us. These cheeses are the collective memory of France. He has been a friend ever since.
Alphonse is a young man who loves his region passionately. His family has made cheese there for many generations. And they use caves that were dug in volcanic rock over 200 years ago. And when I first visited his cave, really, it is the most beautiful cave that I have seen. Because a cheesemaker used a cave that is built into the earth, has been there for centuries. There are strains of fungi that developed, and the cheesemaker saw, if I keep the cave at this temperature, or if I add rice straw, this gives a great cheese. The first goal of my work in France was to collect natural local strains of the fungus called Geotricum candidum, and then to test the strains I collected, or part of them, for diversity.
So we use the genetic technique of amplifying DNA, and then we were testing to see these fingerprints to see if the isolates I collected were individual different strains of Geotricum candidum. So these are 18 isolates from 7 facilities, some as close as 2.5 kilometers to each other. And out of that, 13 of the 18 isolates were unique strains. So we might say, well, why do we care? Well, it's because of this pool of biodiversity, which was really utilized by traditional cheesemakers who saw empirically that if I used a certain tool in making the cheese, wood, if I use a certain kind of coagulant, like the lining of the calf's stomach, or if I used a certain temperature, or if I aged the cheese in a certain moisture, that gave the best kind of cheese. And we want to save this biodiversity. It affects, these different strains could affect bitterness, neutralization of the curd. Some of them are known to inhibit Listeria monocytogenes, and some can inhibit mucor. So we owe really this saving of the pool of biodiversity to the traditional cheesemakers. And just to say, now in this cave, also in the oven, they have to use the shelves to age the cheese instead of putting them on the floor. But notice that even in commercial settings, they are sticking rice straw underneath the plastic mat. And you might say, is this just the stubbornness of the people of the Auvergne? But it's based in the history of the region. My master came from Cézallier, which is Celtic for Pays du Seigle, or country of the rye straw. So the cheesemakers age their cheese on rye straw, which I would learn later is that Trichothecium roseum, that's the flower of the molds, that pink mold is actually a pathogen to rye straw. So they would put the cheese on rye straw and Trichothecium roseum would grow naturally. And they found that gave the best cheese. It's highly proteolytic. So they learned empirically what gave the best cheeses. We as scientists then can identify what the fungus was. And now there's a lot more research going on, which is very important, about the biofilms that are on materials like wood. And in testing the Tina from Ragusa and the Gel from Cantal, they are finding that these biofilms in the wood are inhibiting Listeria, Salmonella, even some E. coli, O157H7, and Staphylococcus. So this is the kind of collaboration, I hope, between science and traditional cheesemakers that can save the pool of biodiversity and also protect these wonderful cheeses that are a patrimony of a country. Thank you very much for inviting me. I'm sorry for the technical difficulties.
Molte grazie, molte grazie.
Thank you. Thank you. I think that Patrick Mercier, who is responsible for the Camembert, traditional Camembert Presidium, so shares many of the things that Madre Noella has just said. Patrick Mercier has come to support us in a tough battle because speaking of Camembert, Camembert is in a French icon like Brigitte Bardot. So Camembert is like a French icon. And so the industry tried and is still trying to steal this product from the natural world. And so where do we stand, Patrick?
Chaque fromage a ses particularités. Et bien sûr,
every cheese has some specific characteristics, and of course we want to make a great cheese that is not and that is also healthy, and it's very important, even though the industry has found ways to industrialize this cheese, and now some camemberts have a very trivial and very monotonous monotonous taste and flavors. So I've been working for many, many years, always raw milk cheeses, AOC cheeses. And little by little, I have seen that there was this attempt on the part of the industries to flatten out these tastes. And therefore, we decided to join forces and wage war against that. And we were talking about raw milk. Raw milk is milk that has not been heated up. And even though we comply with these standards, it can be poor from the microbiological standpoint. And for instance, if we make some cheeses, we can use raw milk, but sometimes the taste is pretty flat, or and also refrigeration can have a role. For instance, preserving cheese at 3 degrees, like we see in the industry, the taste is going to be handicapped. So the natural flora is very much affected. And also grazing, pasture grazing is also very important for our raw milk. This means having cows in the fields, and they can graze grass, they can have a lot of microorganisms on this grass, and therefore when they lie on this grass, the cows are going to absorb this biodiversity. And so if we do not refrigerate this milk, and if you do not clean too much the cow's udder, we can preserve this microflora in the final cheeses. But that's a very fragile cheese, because the need to guarantee the absence of pathogens is such that we all try to disinfect and clean the udder, the cow udder. Then we check and test that there are not pathogens. And therefore, in the end, we decided to try to use some common sense and wisdom. For instance, we don't use silage for our animals. We don't refrigerate milk. We don't clean too much the cow's udder. And we keep the milk at certain temperatures, and just allowing microorganisms or bacteria that are in the fields to then convey our notion of terroir to our cheeses. And this is a fight that is very, very tough, because this seems to contradict all what we have always learned, and all the hygienical standards and guarantees of given to consumers. And for our cheese, our camembert, so it's a soft cheese, and which is matured over a short period of time. And in case there is a contamination, there's no chance that it disappears in our production process. And therefore, this is an additional challenge that we encounter, but we are ready to fight. And actually, we have been doing that for 9 years, because we have been making camembert de Normandie for 9 years now. And we have And we are sure that our farm and our cows can convey a very special flavor. And we decided to leave the AOC denomination because we had become so popular that it was no need for us to be called AOC, because we could no longer have that label with our production method. And we add some starter. However, why do we add some starter? Because we need to manage our time. I mean, we have 10 people, and we cannot work more than so longer than, so, or after 8 o'clock in the afternoon, in the evening. And to make a camembert, we have to be present from 6 in the morning to 8 p.m. But if we do not use some yeasts, we would not be able to leave the farm at 8 p.m. So we work with Khan University to grow this microflora to facilitate this process. And of course, we don't disinfect the cow's udder, we just clean them. And this is necessary to do because of the French law. And we hope we can find these additional yeasts in our fields. So that's why now we are studying with the Cannes University these microflora so that we can use that microflora without adding other starters in the future, and ensure and guarantee to our consumers that no microorganism is added. So foreign alien microorganisms are added. There are many recipes for cheeses. There are some easy recipes that you can use to say, well, I don't add anything, it's supernatural. And I think we have chosen, and I'm proud to have chosen, one of the toughest cheeses to make, very difficult recipe, because it's damp, so it's humid, and it's not very lactic, and it preserves a lot of water, so it has a lot of water. So we have all what it takes to make it difficult to make. So if we truly want to make good cheese, and so that's the challenge we've decided to take on.
Voilà. Je crois avoir, j'ai pas suivi mon plan du tout.
I didn't follow the list of topics I wanted to cover at all. And in any case, this was just to convey my passion and describe what we've been trying to do, what we do every day. So feel free to ask me any questions if you like. I have a curiosity. Don't you use whey starters or milk starters? Can you hear me? Well, well, what we try is not to have problems for all our cheeses. So we have this 3-week maturation process, and of course we identify every batch, and adding whey starters would lead us to have a high risk. Maybe we will verify that with Cannes University, so we may use that, but for the time being, we don't dare doing that. So in actually at the moment, nobody in our region uses the whey in this way. Well, okay, I disagree with what you say, but I accept your point. Now I would like to ask Duccio for his remarks. Duccio Cavalieri has been supporting us for many, many years. He's a professor of microbiology at the University of Florence, and he is an expert of complex systems. So what more complex than the microflora in cheeses? Are there methods to protect this invisible biodiversity in a way that is not invasive? I mean, can we control it and in a way that is not too intrusive and not too invasive? I hope you can answer my question. Well, I believe that the first element, if in my answer, is that we need to have a very good knowledge of the biodiversity we have in front of us. And also the experiment that Mother Noella showed proves that actually we know very little about these processes to be able to guide them rationally with our human rationality. And I would like to remind you only a few points about how we as human beings have started to control microbe biodiversity, and cheeses can be a very good example of that. And I wanted to do that without slides, but then I thought that some slides might help us. And therefore, I'd like to share my slides. I'd like to start from these key concept. That is to say, cheeses are part or belong to our cultural evolution and must be seen in the framework of a microbial anthropology. That is to say, how we as humans went through different conditions in our lives, in our evolution, and adapted to various diets, different diets. So to answer your questions, for instance, I could say that only in 1660, so when Antoine van Leeuwenhoek developed the first microscopes, that we can see these structures. And so far, we have seen them in the microscope, through the microscope, or thanks to the great work of Fanny Iselius, who worked with his husband Hesse and Petri. And so we started seeing these microorganisms on the plate. This discovery has never been awarded and acknowledged to this woman, but she decided to use agar-agar to make the Petri dishes. Actually, she could use cheese rind, and probably some people have done that, and actually cheese rind could be an excellent model to use. And now we have evolved quite a lot and went beyond the knowledge on this plate, because on the plate we know there's less than 1% of the microbe biodiversity. So if I take a gram of soil, I know that 1% of those microorganisms can be cultivated. So how can I know? How do I know I have that the other 99%? Because today labs like mine or other labs use the DNA study contained in soils, in fishes, in the human guts, or in a cheese or yogurt to study microbial diversity, biodiversity. And what do we see? Well, we see that microorganisms create man, are man, humans. So we live because there are microorganisms. We contain 2 kilos of microorganisms which interact with our body through 300 square meters of intestines. Since the times of Hippocrates, we know that all diseases come from the intestine, from the guts, and also health comes from the guts. And this microbiota, this microbial community, is something that we share with the external environment. And the diet is one of the fundamental systems to change our microbiota. And I would like to reverse our perspective, our vision, because we evolved initially like insectivores, then as hunters and gatherers, and then until 15,000 years ago, we started to domesticate animals and discovered agriculture. So in this long period of time, we changed significantly our diet, and we started eating foodstuffs that we did not eat earlier. So since the times of hunters and gatherers, animals were killed. And when animals are killed, are domesticated on the contrary, we started using milk from these animals. And this poses a problem. Milk is digestible only in the first years of life, because when we grow up, we are no longer children. The enzyme need to digest milk is no longer produced. And in any case, we coexist. We have coexisted with these animals for 15,000 years. So how have we made it possible for for milk to be preserved? We invented cheeses, and actually the microorganisms of yogurt and cheeses enable people who no longer have the gene to digest milk to be able to digest milk. And most of milk intolerances and food intolerances today result from the fact that these microorganisms that for thousands of years we have absorbed, we using living food with a lot of microorganisms, these microorganisms are disappearing. So I want to reverse your question, because you asked me what can we do, or is it possible to manage these microorganisms and make products without industrial or artificial systems. Well, actually, we have to do that, because these cheeses have acted as a filter to ingest into our body the microorganisms that are very important to keep us healthy. So these are bacteria and yeasts, and cheesemakers for hundreds of years, for centuries. So cheesemakers have always isolated these microorganisms, lactobacilli, lactococci, streptococci, but those used in the industry, and this is work about the production of Parmigiano, mozzarella, and other cheeses, show that what we use in the industry are very few. So we are using really a handful, a very tiny number of microorganisms. So when we, for instance, this work by Rachel Banda, and I've spent 6 years in Harvard, and she's a dear friend. We work together, same institute. And we saw that what Mother Noella said can be tested and studied. And so the biodiversity we have is much higher, much bigger. So we have hundreds of thousands of species of bacteria and fungi that proliferate on the rind of the cheese rind and make this product what it is in the end. I think we simplified over the years because we had to manage complexity, but now we have the tools not only to tackle this, and these DNA studies don't require us to grow anything. In the DNA test, we can see everything, and we also see pathogens. True, there are some pathogens, often in a very small quantity. I worked at San Michele all'Adige, and actually, very occasionally, there was some salmonella on some cheeses, but these microorganisms inhibit the growth of pathogens and salmonella, for instance. I mean, cheese is a process which we've used in our evolution to preserve and eat a product that otherwise would be thrown away, milk. And as to the aromas, I mean, this is a recent review on this topic. And as you can see, the division of bacteria has led to simplification. This homo or hetero fermenting bacteria has led to a flattening of our tastes, because many of the aromatic and flavor characteristics actually come from the bacteria, the hetero fermenting bacteria that the industry is discarding and to simplify production. So back to health. Leah Metchikoff was the inventor of, or actually of the probiotics. Many probiotics, so microorganisms that we buy as supplements at the pharmacy, are present already in our cheeses, in our natural cheeses. My work is conducting studies on microbial anthropology, and we went with some collaborators from Bologna University all the way to Tajikistan to study kefir from Tajikistan, and also we are studying kefir from Romania, and we are trying to study ever different cheeses. And we have actually seen that the amount of strains, different strains and individuals in the production of kefir or cheese is huge. And so why is this important? So why should we communicate to the public that this is important? Because for 80 years, we are confronted with the third big evolution in revolution in our evolution, because we moved from the Neolithic revolution to the revolution of the supermarket. I mean, globalization has led to a huge simplification of production processes, and the globalized diet is right behind many diseases, ATT, AGS, Crohn, etc., which are a huge health problem across the world. And therefore, if we recover the microbial biodiversity, which is part of traditional cheeses, that come from the world that is not yet globalized. This is from the evolutionary medicine school. So we can recover this microbial biodiversity and go back to a world where we don't die of inflammation. And we can take care of our health with our food and our microorganisms, as we've done for some hundreds of years. So my message is that we have the way to characterize this biodiversity. We can do that. We have tools to do that. And you may say, oh, yes, but these tools cannot be moved from the lab to the small dairy farm. But actually, I can tell you that we have used it to study the COVID variants. So today we can bring about this huge technology revolution to study this biodiversity on site. And we can give cheesemakers and producers some tools that can allow them to make products and safe products and very enjoyable products. And therefore, the understanding of microorganisms, more than for making a perfect starter, can actually be useful to become ever more knowledgeable consumers. Thank you, Duccio. Thank you. Thank you. Your thought process is fine. The problem is then to translate all these in practical actions. And we as Slow Food should do that, but it's pretty tough, but we keep going, we insist, we keep going, we don't surrender. So now we have, again, 4 or 5 minutes each for each of you. And I would like to ask Mother Noella if this awareness that there is this invisible world among the consumers who are very fond of hygiene, especially. So do you see a difference in the perception of the consumers, in the producers? So are they aware of this or not? What do you think?
Well, when we first started making our cheese with the fungi on top, we used to have an annual fair, and the Americans that came to the fair were afraid to taste the cheese because it looked dirty. But after tasting the cheese, within 2 years, we would sell every morsel of cheese in the first few hours because they tasted the cheese and loved it. They didn't get sick. So, and I have found that the idea people have of cheese being dirty because there are fungi on it has completely changed in the United States. And I think people are looking for local foods, natural foods, and I think they want to support the small producers. I think there's a great movement in the United States now for these natural products and a sense now of terroir. And I think that, we know the French wrote the book in terms of terroir, but the American cheesemakers, the artisanal cheesemakers, especially the Keelers, the brothers in Greensboro, Vermont, they have a strong sense of terroir. They're naming their cheeses after local historical characters, and they even have a lab. That's where Rachel Dutton worked there for a long time, and Ben Wolfe. And they are testing local microorganisms within their cheese production. So I think things have changed very much in the United States in the last 30 years.
Anche meno.
I would say even less than 30 years, I would say. And in France, there is more awareness. Is that right, Patrick? Even though it's a bit difficult to talk to French people about natural cheeses. What do you think, Patrick? Well, as I said, there are some AOC cheeses that like Comté, and I know this region very well. And I learned a lot there for our cheeses. So let's say that some cheeses lend themselves more to adopt these traditional natural techniques. And there are cheesemakers who are very keen on using these traditional systems. For some cheeses, it's easier. This doesn't mean that we shouldn't do that for all cheeses, but however, it's very important to avert and to avoid health and hygienic problems. Also because if I have one day a problem of this kind, the industry would totally kind of kill me. And so we cannot afford making mistakes. And for instance, we have our cheese called Le Champ Secret, the secret field. And so there are still some challenges to make sure that this is completely natural. Of course, we are very passionate about this work, but we cannot make mistakes because otherwise we could spoil all our reputation if we ever have any problem. So therefore, this caution, and this is a very French way of, so before you were talking about traditional tools. For instance, we use a sieve to drain our camembert, and it's made of wood. And that's very important, but the French administration wanted us to use the plastic sieves, and so we have to thank the European Union for allowing us to use these traditional materials. And for instance, this wooden structure that we use when we use a new one, it takes 3 weeks before we have the same flavors. So actually we use the old one with the new one below, so that the new one can absorb the flavors and the aromas typical of our cheese. And also what is very important is a very diversified diet of our cows. And this is important for the animals and also for us.
Ecco, vorrei chiedere a Duccio.
And now I would like to ask Duccio, so this idea of controlling production, and is it so closely related to hygiene, or is hygiene only a part of the problem? Well, certainly hygiene is only a part of the problem. Let me give you an example. So it was beautiful, talking about fields and herbs and flowers, etc. And think of the impact of chemicals over the last 60, 70 years in our fields. So this is trying to control the ecosystem, but we should be able to control microorganisms, but by considering the entire the ecosystem where they are. For instance, Mr. Mercier said, I changed the microbes by just cooling milk at 4 degrees because they can change the different densities of microorganisms. And therefore, we can think what it means for us. For instance, we in the vineyards, for instance, we use a lot of pesticides and fungicides, but this doesn't only kill the pathogens of the vines in the vineyard, but kills everything in the ecosystem. So this is a complex system and must be regarded as a unicum, as a whole system. And then looking for pathogens in like salmonella, of course, we have to avoid that. We don't want listeria too on cheese, but we should also consider that there is a theory that is at the basis of evolutionary medicine that says that encountering some pathogens when we are children is not bad, because this teaches your immune system to recognize good pathogens from bad pathogens. Our children live in a sterilized world. Look at what is happening with COVID, with these keep continuously cleaning the hands. I am for vaccines and I am for protection and I am for, but I see this is a big catastrophe, but excessive hygiene causes damage to the skin, for instance, and so we shouldn't be too much in extreme positions. For instance, when we were children, probably me and you, we were putting pebbles in the mouth. And for instance, Professor Schlosse studied what was the best way to restore the microbiota of a mouse that was born sterile. And the best thing turned out to be to give soil to eat to the mouse. And for instance, a dog that has diarrhea would eat soil. to get rid of diarrhea. So the soil, the earth, is certainly a key element in biodiversity, which is necessary. Of course, that soil contains good microorganisms and some pathogens, certainly, but certainly positive ones too. And for instance, we've worked in Burkina Faso in Africa, and we saw that the Burkina Faso children were eating in hygienical conditions that were terrible. They were drinking water from a little pond where with all animals all around. The levels of streptococci, fecal Klebsiella and streptococci was less than those of children living in Western countries just eating sugar and candies. And so we are publishing a third paper on this because we've seen that that soil, that earth, so that food, so to speak, contaminated food contained some microbes that acted like natural antibiotics. So bacteria fight with each other, and perhaps cheeses, natural cheeses, contain some bacteria which fight other bacteria and destroy them. We can call it a kind of biologic fight, but we could say that's a self-sustainability of health. Okay, this opens up a lot of topics. We are at the end of our event, and Noella raised your hand. You have 4 minutes. We still have 4 minutes. What do you?
Okay. What I just wanted to address some of the things that Patrick said, because I think it's very important to differentiate the type of cheese we're talking about in terms of safety. It's one thing to talk about a Comté or a Parmesan, which are cheeses with less moisture than Camembert. And a cheese that is drier is safer. So I do feel what Patrick describes he is doing, especially in collaboration with the University of Caen, is very important. And something like Camembert has a high pH because of the fungi, which again would make it more susceptible to pathogens, and with the high moisture content and a shorter ripening period than something again like a Comté or a Parmesan. And I think that's very important for people to know that the safety of the pot mold cheeses is something we need so much research on. We know that many of the ripened cheeses, 60 days or 3 months, are much safer. And I feel I'm very impressed with what Patrick is doing, and especially that he's collaborating with the university. And I understand completely why he's using starters. I think what cheesemakers have to be careful about in adding starters, which I'm for in some instances, is that if you add too much, then there's a lot of competition. There's not much space. They're fighting for space and nutrients. So if you add too many commercial cultures, they can take over and the natural ones can't survive. So I think cheesemakers have to experiment with that. But I just wanted to say, I think the things that Patrick is doing is a way to save a beautiful cheese like Camembert, but also to utilize the natural diversity of his farm.
Ma Patrick ne si è assolto, ma Madre Noella lo ha.
So Madre Noella gave you the absolution, so you absolved him. I do that a little less though, but of course the problem is there. We have to, so it's impossible to get rid of it overnight, but we have to know very clear that then these industrial starters prevail in the end, because they go straight away like a sword, whereas the natural ones are weaker. So if you make a choice, do that up to the end. But of course, I'm not an expert, and I mean, I don't want to criticize Mercier's product. So I hope that in the end you will manage to make natural cheeses. It was all very, very, very interesting. We should have more time to discuss about that. Allow me one last comment. We have to be careful to have certainties about the prevalence of starters, because in winemaking, not in cheese making, but in winemaking, the starter, the use of starters in the end didn't turn out to be the prevalent strain. And it would be very interesting to study, but I think that probably Patrick is doing that, to what extent the starter strain is going to change the global community. I agree with what was said earlier, that is to say, acidity is a key element, and also maturation is a key also for safety. So because one way to inhibit pathogens is to increase lactic acid, so acidity. And so it's true that the camembert in some cheeses that are hard to control from the microbiological standpoint. And so I understand why you use starters in the end, because I mean, we are walking on a thin line, you know, so we are on a sort of blurred boundary between safety and no safety. However, I think that here we need more studies, and these would be wonderful ideas, wonderful models that we should investigate more with the universities. Okay, but let me raise another question, but don't answer. 2 centuries ago, there were no starters. So did people die all over the place? No. And no, they were dying because of something else, something else. Okay, but now we have no time to answer. Often they were dying of hunger, so they were starving, actually. In any case, thank you all. Thank you all. Enjoy your evening. Thank you.